Pyruvate oxidation is the second step in cellular respiration. Following glycolysis, we have produced these pyruvate molecules, 2 pyruvate molecules produced from each molecule of glucose that went through glycolysis. This pyruvate molecule, of which we have 2, is going to then be oxidized in this pyruvate oxidation reaction, sometimes called a transition reaction. So this occurs in the matrix of the mitochondria. So this is the innermost part. Remember, the mitochondria has 2 cell membranes. Cell membrane is probably the wrong way to say it, but 2 phospholipid bilayers. It has a outer membrane, as shown here, and an inner membrane, as if it were made from a cell being engulfed by another cell, hence our endosymbiotic theory. But anyways, the inner part of this mitochondria, that is the matrix, and this is where pyruvate oxidation is taking place. So the 2 pyruvates that are made from the 1 glucose molecule that went through glycolysis, and again, the glycolysis is happening in the cytosol. It has converted the glucose molecule into pyruvate through the 10 steps of glycolysis, and we now have this pyruvate molecule that gets transported into the matrix of the mitochondria, having to go past 2 phospholipid bilayers, the outer membrane and the inner membrane, and now it is loaded into the matrix. And again, so this is the matrix of the mitochondria. While it is there, it is able to go through this process of conversion into acetyl-CoA. So the first thing that it does is, you'll notice that it is, wrong circle there, it is a 3-carbon molecule. Acetyl-CoA is a 2-carbon molecule because one of the steps in the process is removing carbon dioxide from there. And so you're breathing out carbon dioxide. This is the first place where that carbon dioxide is generated in cellular respiration. So this carbon dioxide is coming off of the pyruvate. The other thing that is happening is electrons are coming off as well, as NAD picks them up and becomes NADH. So the pyruvate has lost carbon dioxide and it has also lost electrons. It will gain an acetyl-CoA molecule, and then we have, this is the acetyl part of it. And sorry, I said acetyl-CoA. It'll gain a coenzyme A, and it'll form this acetyl-CoA part. And this is the this double carbon here. The acetyl group is the remaining part of the pyruvate, and it is now attached to a CoA, and normally we just write CoA as the name for this molecule, acetyl-CoA. So the carboxyl end is removed as carbon dioxide. So again, let's just highlight that particular part here. So this carboxyl end, and again, it's the hydrogen removed currently, it is going to actually come unattached, and that is where we get our carbon dioxide from. It gets oxidized. Remember, oil rig, oxidation is loss of electrons. So the pyruvate is oxidized. It is giving up electrons. You can imagine electron is coming off of this pyruvate. Pyruvate is losing it, and NAD plus is gaining. Reduction is the gain of electrons. So the NAD plus is gaining electrons, and now we have this fully charged electron carrier NADH. That's being formed here. And then coenzyme A is being added to make acetyl-CoA. Realize that the acetyl-CoA is going to come with the hydrogen attached. Pyruvate may have this hydroxide instead of this OH on it. So depending on which diagrams you're looking at, you may see it with the hydrogen or without the hydrogen. Again, our ongoing neglect of hydrogen in biology continues. The CoA is a pretty short name to write, and here is why. So again, the acetyl-CoA is just the acetyl group is really just the 2 carbons there with the double bonded O, and then all of this is the CoA. So this is why we just write CoA. It actually does become a much larger molecule as it becomes acetyl-CoA, but that's the CoA enzyme part, if you're ever wondering. That looks familiar. All right, so in summary, we went into this with 2 pyruvates. We had our acetyl-CoA and we had our empty electron carriers. The electron carriers became filled, they got reduced, they gained electrons, and now we have NADHs. And remember, this is happening for 2 pyruvates for every single glucose that started into cellular respiration. The CoA is now attached to the pyruvate, and that gives us our acetyl-CoA molecule, which is the one part that is going to continue the process into the citric acid cycle. 2 carbon dioxides have come off, and this is the beginning production. Remember, the overall process produces 6 carbon dioxide. Here's 2 of them accounted for. And then there's some things going on with hydrogen that we don't care to track. This CoA part that we do produce, we're going to follow it along into the Krebs cycle, or citric acid cycle. The NADHs, remember we saw them earlier with glycolysis as well. So we have produced 2 more of those guys as well. We're going to be saving them, putting them aside, and using them at the end, the 4th step, during the electron transport chain. And the carbon dioxide produced in the matrix there is going to have to diffuse out through the double phospholipid bilayers, the 2 of them, in the mitochondria, and then out through the cell, and eventually expelled as a waste product. Coenzyme A is an important molecule, so this is the ending point of pyruvate oxidation. But if you look at where it fits in with some other metabolic processes, you can see that you can make it from things like glucose, as we've been tracking, but also proteins and lipids, or other macromolecules, can also be broken down for energy, and they get to the point of acetyl-CoA as well. If there is not enough ATP in the cell, if there's not enough, we're going to be using that acetyl-CoA to make some energy because we need more. If there is, I read that carefully, is sufficient ATP, then we can actually reverse some of these processes, and we can take this acetyl-CoA and turn it into long-term storage as lipids. So think of this as a crossroad. We have a meeting point, and acetyl-CoA is a very important molecule. It's like the junction point, crossroads, for several different metabolic processes. Here it is here. So we are focusing on carbohydrate metabolism, doing our glycolysis portion here, pyruvate oxidation, that gets us to acetyl-CoA. But we know that we can get energy from proteins, and it also is going to stop at acetyl-CoA at some point. We get energy from fats and lipids, and it is also going to get to acetyl-CoA at some point. And so this is a very important crossroads for several metabolic processes, all of which can be used to form energy. And realize we can connect this, so you can take your carbohydrates and you can turn them into things like fats by using that acetyl-CoA as an intermediate as well. Citric acid cycle is what we'll look at next, and then we'll finally end with what's called the electron transport chain.